So, yes, very good morning. Today we are going to deal with the chapter anatomy of flowering plants. Anatomy of flowering plants, most of the students will feel this chapter as lengthy, but to learn this chapter in a smart way, just fragment this chapter into 3 fragments. Okay? First is tissues, second segment is tissue systems, and third segment is learning dicot and monocot comparatively, okay, dicot and monocot comparatively, all for root, stem, leaf, very good. And of course, we all know that from 2024 secondary growth is being deleted, but you have to learn secondary growth in the context, okay. Finally, secondary growth you have to learn in the context of differentiation, de-differentiation and redifferentiation. Okay, what is it beta? Differentiation, de-differentiation and redifferentiation. In this context, you have to go through the secondary growth, okay. And this differentiation, de-differentiation, redifferentiation, this concept of anatomy is connected with one more chapter in our NCERT, plant growth and development. This is how you have to deal with the chapter, okay. First into 3 segments beta, what is that? Tissues, tissue systems and dicot, monocot, root, stem, leaf. And secondary growth, in what context you have to learn? Differentiation, de-differentiation, redifferentiation, which is connected with plant growth and development. So, first if we have to look into the basic definition of tissues, what do you call tissues as? Tissues are group of cells, okay, which are functionally similar and which has same origin, okay. Tissues are functionally similar and all the cells in the tissue will have same origin, but they can be structurally similar or dissimilar, okay. They can be structurally similar or dissimilar. Clear beta? Okay. What do you call tissues as? Group of cells which can be structurally similar or dissimilar, but they are functionally similar and all those group of cells have same origin. Clear, right? The primary differentiation of tissues is based on the division capacity or not, okay. The primarily tissues are divided into 2 sets based on the capability of division. Based on what beta? Capability of division. Fine. Based on the capability of division, primarily tissues are divided into 2 things. What are they? Meristematic tissues. These tissues will divide continuously, okay. Permanent tissues, they will not have any capacity of division, okay. They exhibit functional differentiation. What do they exhibit? Functional and structural differentiation. Structural and functional differentiation. Clear? So, yes, how do you define tissues as? Group of cells which have same origin, functional similarity, structurally similar or dissimilar. Initially they are divided into meristematic and permanent only based on the capability of division. Meristematic tissues will continuously divide, permanent tissues lost their capacity of division and they exhibit structural and functional differentiation. Now, if you have to see meristematic tissues beta, based on the position, it is divided into apical meristem and intercalary meristem. So, yes, if you directly take a shoot, okay, at the nodal regions you can find out the branches like this, yes or no. The Meristematic tissues which are present at the apical region of the shoot and the root. These are called as what? Apical meristems. And those meristems which are present in between two nodes, these are called as what? Intercalary meristems. Clear beta? Apical meristems and intercalary meristems. And what are secondary or lateral meristems? Those which origin in the later part of plant life. Okay. Once the plant has finished its primary growth, okay, secondary meristems or lateral meristems, they will appear in the later part of plant life. Fine. Wherein you have two types of secondary or lateral meristems. They are vascular cambium and cork cambium. Vascular cambium is present in the stele region. I will explain you clearly what is stele region. It is present where? In the stele region. Whereas cork cambium, where is it present beta? In the cortex region. In the cortex region. Both of them will aid in what? Secondary growth. Fine. The primary meristems will build up the primary plant body. Secondary meristems or lateral meristems will increase the girth of the plant body. Clear? Understood these two? Fine now beta. Next, permanent tissues. If all the cells in the tissues are structurally similar, okay, if all the cells of the tissues are structurally similar, then you will call it as simple tissues. Okay, if all the cells in this are structurally similar, you will call it as simple permanent tissues. They are of three types: Parenchyma, Collenchyma and Sclerenchyma. Complex tissues are of two types. They are what? Xylem and Phloem. Here you can find the structural differentiation. For example, xylem is composed of four tissues. What are they? You just tell me. In angiosperms, xylem is characterized to tracheids, vessels, xylem fibers, xylem parenchyma. Yes or no? Phloem is also differentiated into sieve tubes, companion cells, phloem fibers and phloem parenchyma. Understood clearly? So, is complete tissue concept aware of you? Have you got the framework of complete tissues? Tissues, do remember, again I am telling you, they can be structurally similar or dissimilar, but functionally they are similar and they have common origin. And based on the capability of division, initially they are differentiated to meristematic and permanent. Meristematic based on their origin, okay, whether it is in the primary plant body or secondary plant body, it is differentiated into primary meristems and secondary meristems. Primary meristems again based on the position, apical and intercalary, okay. Secondary meristems based on the location, position again. Vascular in the stellar region, cork cambium in the cortex region. Completely aware of the meristematic tissues, right? Permanent tissues based on the cells which are structurally similar or dissimilar. You have simple permanent tissues as Parenchyma, Collenchyma and Sclerenchyma. Complex permanent tissues as xylem and phloem. These are the set of tissues which are present in angiosperms. Let us get into the detailed study of meristematic tissues first. How you can remember a meristematic tissues? Meristematic tissues means what beta? Those cells which completely divide all the time, right? Their only activity is what? Division. If cell needs to divide all the time, its cell wall should be thick or thin? Thin, right? Its cell wall should be thin and it should have conspicuous nucleus, means very small nucleus, and dense cytoplasm. Dense cytoplasm because it is dividing continuously, and they have small and immature cells. All the cells we can divide through the same plane, that is why we can say they are isodiametric, okay, and few at times they are of different shapes like round, oval and polygonal, isodiametric. Next, vacuoles are totally absent because there is no necessity of vacuole only because they are dividing continuously. Next, rate of respiration will be high because of division activity. Because of what? Cell division. See, here there is nothing to mug up beta. There is nothing to mug up. Just think logically. The cells are dividing continuously, means cell wall will be thin. Conspicuous nucleus will be there. Vacuoles will be absent or small. Okay, cytoplasm will be dense and all cells are isodiametric. Because of the continuous cell division activity, they show high respiration rate. Clear? So, these are the characteristic features of meristematic tissue. Understood, Navita? Next. So, this is the diagram which is given in your NCERT. Okay, we can expect diagram based question also from here. So, look properly. This is what? Root apical meristem, and this is what? Shoot apical meristem, right Nandu? Root apical meristem and shoot apical meristem. Root apical meristem at the terminal region or at the final region. This is called as apical or terminal region. You have what? Root cap. Root cap is present where beta? Apical or terminal region. Fine. In the subterminal region, you have root apical meristem. Root apical meristem is present where in the subterminal region. Fine. And see, close to the root apical meristems, you have initials of central cylinder and cortex. From this regions, cortex will be developed from this. Okay. The cells which are in close proximity to the root apical meristem, they have what? Initials of central cylinder and cortex. And the outer layer of this shoot or the root apical region, outer layer is what? Protoderm, which is future epidermis. Okay. And the central parts you can see clearly, central cylinder and cortex, coming from the initials of central cylinder and cortex. Central cylinder means what, Nandu? It is vascular cane. Clear with the root apical meristem structure? Fine. You need not to mug up labelings at all. Okay. Understand it properly. Understand the diagram and the label which is given, then it will fix in your mind automatically. No efforts to remember it too much. Next, same case if you identify with root apical meristem. Root apical meristem is present at the apical region. Sorry, shoot apical meristem is present at the apical region, not like root apical meristem. Root apical is at subterminal or subapical, but this is at the apical region. Okay, shoot apical meristem is at the apical region. And if you identify, if you observe, one second. So, the angle which is formed by the leaf and the stem, this angle is called as what? Axil, right? The angle which is formed between leaf and the stem is called as what? Axil. In the axil region, bud will be developed, you will call it as axillary bud. See, shoot apical meristem and here is the axillary bud. And to the top region of shoot apical meristem, you have leaf primordia. Shoot apical meristem, tip of the region you have leaf primordia, the angle between this you have axillary bud, and at the center you have again central cylinder, or you can also call it as differentiating vascular tissue. Clear? Understood clearly the diagrams of root apical meristem and shoot apical meristem? Clear, Nanandu? Yes. Next, shoot apical meristem and root apical meristem. So, in this if you have to see all this shoot apical meristems, you have two types, shoot apical and root apical, whose only activity is what? Division. And intercalary meristem is present where, ma? Intercalary meristem is present in between two nodes, two nodal regions, intercalary meristem is present. Intercalary meristem is mainly responsible for increase in length of the stem, increase in length of the stem. And whenever the grazing herbivores eat the grasses, okay, the apical meristems will be cut off, right, but still you can see the growth of the plant because of what? Intercalary meristems. That is a point given in our NCERT, just do remember. This is all about intercalary meristem, apical meristem. These are what? Primary meristems, primary meristematic tissues. Clear, ma? Now, secondary meristematic tissues are also called as what? Collenchyma or just a second. It is called as, secondary meristems are also called as lateral meristems. It is of two types. What are they, beta? Yeah, vascular cambium and cork cambium. Okay. For example, I am taking for example stele of dicot stem, let us assume. In dicot stem, what will be the xylem vascular bundle arrangement? It will be conjoint, right? Okay, vascular bundle arrangement will be conjoint. Fine. Xylem at the center, phloem towards the periphery. This type of vascular bundle arrangement is what? Conjoint. Why is it conjoint? Xylem and phloem are at the same radius, right? That is why it is called as conjoint. Cambium is present first only, so it is called as open, conjoint open. And two vascular bundles are lateral to each other, it is called as what? Collateral. So, in a dicot stem, what will be the vascular bundle arrangement, ma? Conjoint, because xylem and phloem are at the same radii, okay, and open because there is a cambium between xylem and phloem, and then collateral, okay, where two vascular bundles are lateral to each other. Understood? Okay, conjoint, open and collateral. So, here Nandu, you see the cambium is present from the beginning only, that is why you are calling it as open type, right? Cambium is present between in between xylem and phloem from the beginning itself. So, this is primary in origin, okay, this is what? Primary in origin. When it is primary in origin, you cannot call it as secondary meristem, right? The cambium which is present in between xylem and phloem, this is called as intravesicular cambium. Intravesicular cambium. Fine, this is present in between xylem and phloem. So, this is primary in origin. Intravesicular cambium is primary in origin, remember that. Next, intervesicular cambium, which is present in between two vascular bundles like this. Okay, intervesicular cambium, what beta? It is present in between two vascular bundles. This is secondary in origin. This is secondary in origin. Now, you can call it as a secondary meristem. So, in vascular cambium, what is secondary in origin? Intervesicular cambium. Okay, what is secondary in origin, ma? Intervesicular cambium. Okay, understood, Nandu? Clear, right? So, in stele, you will find two types of cambium, two types of vascular cambium of dicot stem. The cambium which is present in between xylem and phloem is called as which one? Okay, that is present in between xylem and phloem, which is of what origin? Okay, and the one which is present in between two vascular bundles, which is of what origin? Secondary in origin. Clear, right? Now, cork cambium is present where? In the cortex region. So, in the cortex region, if you assume the cork cambium, which is also called as phellogen, it will divide and form new cells towards the epidermis. You will call it as cork cells, and new cells which are formed towards the endodermis are called as what? Cork cambium. You will call it as what, ma? That's it. You have understood, got a clear idea about the secondary meristems, both the vascular cambium and cork cambium. Clear, right, ma? Next, online students, any doubt anywhere you can ask me, beta. Now, coming to permanent tissues, in the beginning slide only we have learnt that permanent tissues, if they are having similar structural cells or structurally dissimilar cells, you have two types. Okay. So, permanent tissues, you have two types of permanent tissues. You have simple permanent tissues and complex permanent tissues. Clear beta? Simple permanent tissues you tell me Nandu. Yes, parenchyma. Yes, very good. Parenchyma, collenchyma and sclerenchyma. Of these three simple permanent tissues, tell me which tissue is most abundant in the plant body? Think and say. Online students also. Of these three permanent tissues, which tissue is most abundant in the plant body? Complex permanent tissue is of two types. Yes, xylem and phloem. Xylem you have tracheids, vessels, xylem fibers and xylem parenchyma. Phloem you have sieve tubes, good, and very good and very good. Okay, so these are the four tissues. Remember that. So this set of tissues you will see only in angiosperms. There are again exceptions for pteridophytes and gymnosperms, so we will talk about it. So this thing is clear to you, Noma. Now let us learn each and every tissue in detail. Okay. First is parenchyma. Cells of parenchyma are isodiametric, where you can divide it in equal planes. All the cells have equal dimensions. So that is isodiametric. All the cells of parenchyma are what ma? Isodiametric, and they can be spherical, oval, round, polygonal or elongated. Parenchymatous cells will have thin cell wall, that is cellulosic cell wall. Okay. They can have intercellular spaces and internally they are enclosed by large central vacuole. You can see here. Okay. Internally they have large central vacuole, and peripheral cytoplasm is present. In the peripheral cytoplasm only they have elongated nucleus. So can you see how can you describe parenchyma? Parenchyma have thin cellulosic cell walls. They can be round, polygonal or oval, and they are isodiametric. They have large central vacuole with peripheral cytoplasm and an elongated nucleus. Understood? Easy, right? Now, types of parenchyma. Okay. First, if you see types of parenchyma, you have two types of parenchyma. If parenchyma has chloroplast inside it, you will call it as Sclerenchyma. Okay, which does the function of photosynthesis. If parenchyma which is present in the aquatic plants, okay, of course we know that the most of the plant body is occupied by parenchymatous tissue. In aquatic plants they have large air spaces. Okay, what do they have ma? Large air spaces in between, which you will call it as air spaces. Those are Parenchyma. This type of parenchymatous tissue which has large air spaces, you will call it as what? Parenchyma. Clear, Nama? Yes? Now, functions include this stores the food and provides turgidity to the softer parts, protection of the epidermis, because epidermal cells are also composed of parenchymatous tissue. Formation of water absorbing epiblema. Epiblema you will see it in roots. Okay. And then lateral conduction in xylem and phloem. In the stellar region just now we have learnt, you have collateral vascular bundles like this, right? Okay. They have lateral conduction. Okay. These cells are filled with parenchymatous cells only. So, which is promoting lateral conduction in between vascular bundles? Parenchyma. Okay. Parenchyma is promoting the lateral conduction between xylem and phloem. Next, photosynthesis, if it is Sclerenchyma, buoyancy of aquatic plants in form of Parenchyma. These are the functions of parenchyma. Next, coming to Collenchyma. Collenchyma is also living and it has vacuole inside it, but the cell wall composition is different. Okay. It has cellulose, hemicellulose and pectin. Okay. Collenchyma cell wall will have what? Cellulose, hemicellulose and pectin. And in parenchyma you can observe small intercellular spaces. Collenchyma there are no intercellular spaces. and most importantly they will provide flexibility or the strength to petioles to carry the leaves. That is the major function of Collenchyma, and all the cells they are found either as homogeneous layer or in the form of patches, and intercellular spaces are absent. What is the cell wall composition? Cellulose, hemicellulose and pectin. Now if you see functions now, okay, they provide functions like mechanical support, okay. First is what? Mechanical support by supporting young stem and petiole. Next is what? They provide flexibility to the organs without bending. Next, it prevents tearing of leaves, fine, because the cells of this have strong cell walls, they prevent what? Tearing of leaves. It helps in growth and elongation of the organ also. Now if you see Sclerenchyma, okay, Sclerenchyma, it occurs in hypodermis, vascular region, cortex, stem and leaves. Structure includes Sclerenchyma consists of long narrow cells, and Sclerenchyma has thick and lignified cell walls. Sclerenchyma cells are dead, okay, they do not have conspicuous nuclei inside it, these cells are what? Dead, and they do not have any protoplast. You have two types of Sclerenchyma based on the lumen present inside it, okay. So those are fibers and sclerites. This is a clear differentiation between fiber and sclerite. How a fiber looks like? Elongated, thick walled. This sclerites will look spherical and cylindrical. Cell condition, both of them are dead cells. Lumen is narrow here, and here lumen is very narrow than sclerites, because it is completely filled by depositions of the thick lignified cell wall. Lumen is very narrow. Here you can see a lumen is narrow, but not as that narrow as sclerite. Understood, ma? Next. Yes. So here if you see occurrence, where does the sclerenchymatous tissue occur? In groups in various parts of the plants, and they are scattered in specific parts. Sclerites are scattered in specific parts. And functions, they provide mechanical support. Where are they present specifically? They are found in the fruit wall of nuts, pulp of guava and sapota, seed coat of legumes and on tea leaves, okay. And if you see the main functions of Sclerenchyma is to provide mechanical support. So the common function of Collenchyma and Sclerenchyma is what? To provide mechanical support. Sclerites provide strength and seed coverings. Sclerites form stony endocarp of fruits called as stone fruits, for example almond, fine. So this is all about simple permanent tissues. Looking upon complex permanent tissues, I will not go with much details explaining all this, but I will be telling you one thing. Yeah. So if you see complex permanent tissues, you have xylem and phloem. See, this is the complete tissue of xylem beta, and this is the tissue of phloem. Look into this diagram properly, you will get the complete information. So at the center you can see tracheids, okay, at the center you can see what? Tracheids. Tracheids are present in all the vascular plants. All the vascular plants in the sense pteridophytes, gymnosperms and angiosperms. That is why we call pteridophytes and gymnosperms also as tracheophytes, because they are having what? Tracheids. Tracheids will have, see, elongated tube like structures with tapering ends, and at the center they have pits to conduct the water. Second type of tissue, if you can observe, are vessels. Vessels have thick perforation plates. What do vessels have beta? Thick perforation plates. In a tracheid, single cell you have many cells inside it, but a vessel, one tube is equal to one cell, and they have thick perforation plates. They are cylindrical, these are elongated, fine. Vessels are not present in all the vascular plants. Vessels are the characteristic feature of only angiosperms, This is important point, ma. Vessels are present only in the angiosperms. It is absent in pteridophytes and gymnosperms. In gymnosperms though, there are only 2 exceptional cases. In them you can see vessels. Those include neetum and in pteridophytes of Selaginella. You can see vessels. Important exceptional cases, please take down. Okay. Next is xylem fibers and xylem parenchyma. What are next, ma? Xylem fibers which are dead again and xylem parenchyma. Xylem parenchyma is the only living tissue in the xylem. Okay. This xylem parenchyma will be helpful in what? Storage of food. This is helpful in storage of food. Now tell me, list out all the 4 tissues of xylem. First is tracheids, which are elongated tapering ends and they have pits and pit fields in them. Vessels, if you see, they are long and cylindrical, at the ends they have perforation plates. The conduction of water, when you see comparatively between tracheid and vessel, vessels will conduct more water than tracheids. Next, vessels are not present in all the vascular plants. They are the characteristic feature of what? Angiosperms. In pteridophytes and gymnosperms, they are absent. 2 exceptions, what are they, Nana? Neetum and Selaginella. In them, vessels are present. Next, xylem fibers are dead. Xylem parenchyma is living. Storage. Its main function is what? To store. Next, phloem, if you see. Phloem is also composed of 4 tissues, right? What are they, you tell me? First is, yeah, see, this space is what? Sieve tubes, and at the end they have what, ma? Sieve plates. These are what? Sieve plates, and this is what? Sieve tube. Sieve tube has what? Sieve cells. Sieve tube has sieve cells. These sieve cells are enucleated. Sieve cells have what? Enucleated. They do not have nucleus. Okay, sieve cells inside them does not have any nucleus. So, they are what? Enucleated. But the as it is enucleated, the function of sieve tube is carried out by what? Companion cells. Functions of sieve tubes carried out by companion cells. Okay. That is why companion cells and sieve tube cells are called as what? Sister cells. They are called as what, ma? Sister cells. Fine. Next, sieve tube cells, companion cells is done. Next, 2 more phloem tissues are what? Phloem fibers and phloem parenchyma. Phloem fibers and phloem parenchyma. Clear, Nanandu? So, these are 4 tissues of xylem and 4 tissues of phloem. I have given you the complete gist of it. Understood clearly, right? Yes. So, this is all about this, and here also there is one exception with respect to gymnosperms. In gymnosperms, you do not have companion cells and sieve tube cells. Rather, in gymnosperms, you have sieve cells, and in the place of companion cells, what do you have? Albuminous cells. Remember these exceptional cases, most important. Okay, done. Next, if you have to see, done, clear with all complex permanent tissues, right? Yeah. So, now next we are going to discuss about the protoxylem and metaxylem, or exarch form of xylem and endarch form, or exarch and endarch form of vascular tissues arrangement. Let me tell you vascular tissues arrangement in root and in stem. Okay, vascular tissues arrangement in root and stem. In root, the vascular tissues are arranged in radial pattern. Radial means what? Phloem is arranged to the periphery like this, and xylem is at the center like this. okay, xylem here and phloem is over here. Are they present at the same radius? No. So, that is why the arrangement is called as what? Radial, okay. And the tissue which is present in between xylem and phloem is called as conjunctive tissue. So, in dicot root, secondary growth, how is it happening? In between xylem and phloem, secondary growth will happen like this. This is called as what cambium? Wavy cambium. Understood? Clear? So, this is the arrangement in the root. So, the xylem, when it is forming, xylem cells will be formed like this, right? Okay. So, the first formed xylem is called as protoxylem. What do you call the first formed xylem as? Protoxylem. Protoxylem is towards the periphery and metaxylem is towards what? Center. This type of arrangement is called as what? Exarch. Protoxylem towards periphery, metaxylem towards the center. That type of arrangement is called as what, ma? Exarch arrangement. Fine. This you will see where in the root. In shoot, you have to tell me what is the arrangement? Conjoint and collateral, right? So, in this, how the xylem and phloem are formed? The protoxylem is towards the center, metaxylem is towards the periphery. Okay. Metaxylem is towards the periphery, protoxylem is towards the center. Fine, Nandu, understood? Okay. Protoxylem is towards the center, metaxylem is towards the periphery. So, here you can see meta towards center, proto towards periphery, that is exarch in roots. Fine. Metaxylem towards the center and protoxylem towards the periphery. Sorry. So, this is stem, right? Correct only. Clear? So, in root, what is the type of vascular bundle arrangement? Exarch. Okay. Protoxylem is towards periphery, metaxylem is towards center. In stem, meta is towards periphery, proto is towards the center. Understood? Now, we will see only one comparative study between dicot and monocot stem. Okay. Root and leaf we will deal in the advanced classes. Just look into how you have to learn this. Okay. Dicot stem is a very big structure. So, we will go with the monocot and dicot, both we will go through. First is epidermis. Okay. The tissue is first formed with what, ma? Epidermis. Epidermis is what? Parenchymatous. We have discussed, right? Epidermis is parenchymatous. So, below epidermis, general cortex is divided into hypodermis, general cortex and endodermis. Hypodermis of dicot stem will have what tissue? Collenchyma. Okay. General cortex is again parenchymatous, endodermis is filled with starch sheath. Okay. Now, if you see, next is pericycle. Pericycle is having sclerenchymatous patches. What patches, ma? Sclerenchymatous patches. And the story below the pericycle you know already. Okay. Stele includes what? Pericycle, vascular bundles, and at the center medulla or pith. Okay. Pericycle, vascular bundles, medulla or pith will include what? Stele. Stele includes pericycle, vascular bundles, pith. Fine. So, the tissue composition is clear to you, right? Okay. Epidermis is composed of what? General cortex is composed of hypodermis, general cortex, endodermis. Hypodermis is made up of general cortex, parenchyma. Endodermis is having starch sheath. Sclerenchymatous patches you see in the pericycle. Okay. And what type of vascular bundle arrangement you see in a dicot stem? Conjoint, open, collateral. Okay. Monocot stem is very simple, ma. Epidermis, you know, it is made up of parenchyma only. Below that, hypodermis here is having sclerenchymatous patches. What does it have? Sclerenchymatous patches. Here it is having collenchyma in dicot stem. Monocot stem, what is it having? Sclerenchymatous patches. And next, completely it is filled with parenchymatous cells only. After hypodermis, it is completely filled with what? Parenchymatous cells, or you can say parenchymatous general cortex. There is no division of endodermis, pericycle, medulla, medullary rays, nothing are present. After hypodermis, completely it is what? Parenchymatous. In this parenchymatous tissue, vascular bundles are originated like this. Okay. In this parenchymatous tissue, what is happening, ma? Vascular bundles are originated in the form of patches. Okay. In the form of patches, vascular bundles are originated. This type of origin is called as schizolysigenous in origin. Schizolysigenous in origin. Clear, Nandu? Understood, right? So, this is schizolysigenous in origin. So, understood the differentiation between dicot and monocot, right? So, now we will exercise few objective, and then I will also tell you the concept of differentiation, de-differentiation and redifferentiation. From the concept, whichever we are dealt today is just 60% of anatomy. Okay. I have covered the concepts which have included in the PYQs also. Just answer these questions. Diagram based. Online students also, ma, just go through. Identify the region, Nandu, first. This is what? Shoot apical meristem, very good. This is what? Leaf primordia. This is what? Very good. And this is differentiating vascular tissue. Now identify the option. So C is auxiliary but good, A is leaf primordia, D is differentiating vascular tissue, B is shoot apical meristem in the meristematic zone. Right now? Answer this NEET 2025 question. Monocot stem we have gone through, right? Vascular bundles are conjoint and closed. In dicot stem it is conjoint and open. Next. No. And next. Wrong. Hypodermis is what? Very good. It is sclerenchymatous. So, what is the option, ma? 3, right? Next. Bulliform cells, you will find it in monocot leaf. Yeah, is responsible for inward curling of reef in the flaccid conditions. I will explain you this more in detail, but as of now we are able to identify, right? Good. Next, leaf structure. Online students also try to answer. First you identify the tissues. Okay, this is what? Epidermal tissue. okay, and these tissues are what? Subsidiary cells, okay. These are what? Guard cells, and this is stomatal pore. Guard cells will have outer thin wall and inner thicker wall, right? That is why they are able to open and close. So, here what will be the answer? Next, this question you just look into. Identify the symbol tissue from the following. Symbol tissue, parenchyma, good, okay. So, this question you can answer with by knowing the concept of differentiation, redifferentiation and redifferentiation. So, first you tell me what are the meristematic tissues? Meristematic tissues, which are primary meristematic tissues. What are they, beta? Primary meristematic tissues? Apical meristem and intercalary meristem, right? Apical meristem is of two types. What are they? Shoot apical meristem and root apical meristem. They undergo continuous division and they will form the permanent tissues, right? So, those permanent tissues we will call it as differentiated tissues. So, what are differentiated tissues? Permanent tissues, right ma? Simple permanent tissues and complex permanent tissues. Simple permanent tissues include Parenchyma, Collenchyma and Sclerenchyma. Complex permanent tissues include Xylem and Phloem. Clear with the differentiated tissues? Amongst these differentiated tissues, vascular cambium is formed between xylem and phloem, and cork cambium is formed in the cortex region. Cork cambium is formed where? In the cortex region. So, these cells are formed from where? Permanent tissues only, no? Differentiated tissues only. Few of them are transformed into vascular cambium and cork cambium. Again, what is the function of vascular cambium and cork cambium? Division. What is the function of vascular cambium and cork cambium? Division. So, they are getting de-differentiated, right? Both of them are getting de-differentiated. De-differentiated. Fine. So, they divide new cells and they will form again differentiated cells only, like cork cambium is forming cork cells and secondary cortex. Okay. Vascular cambium is dividing and forming what? Secondary xylem and secondary phloem. So, all this cork, secondary cortex, secondary xylem, secondary phloem, these are what? Redifferentiated. What are they, beta? Understood, okay? So, all this are what? Redifferentiated. Once you clearly analyze, firstly meristematic tissues, they will divide and they form permanent tissues, you will call it as differentiated tissues. Of them few get again meristematic activity, that is why you call it as what? De-differentiated, okay. Again they will undergo division, form permanent tissues. Those permanent tissues are what? Cork, secondary cortex, secondary xylem and secondary phloem. They are what? Redifferentiated. Understood, Nandu? Fine, right? Okay. You have clear idea about meristematic, differentiated, de-differentiated and redifferentiated. Now answer this question, an application of this based on tissue culture. Ma? Look into it properly. In a tissue culture experiment, leaf mesophyll cells are put into culture medium to form callus. So, mesophyll cells are differentiated, yes or no? when they are put into the culture medium, it is giving out callus, means what? It is dividing. Yes or no? A permanent tissue dividing means it lost the capacity of differentiation. It again gained back the capacity of what? Division. Then we will call it as what? That's it. Clear? Understood? So, whatever we have dealt today with respect to anatomy is just 50 to 60%, okay, and this is a strong foundation if you want to learn anatomy more in detailed way. Okay, if you go through this only, you will understand next level tissue systems, what is epidermal tissue system, ground tissue system, vascular tissue system. If you understand this only, you can clear out the clear cut comparative study between dicot, monocot, stem, root and leaf, and you can clearly understand this differentiation, dedifferentiation and redifferentiation concept in a more detailed way. Okay, so it is very important, but it is very important to know this class. Fine. Yes, thank you. So, tomorrow we will come up with a new topic again.